Which grade are you in?
Year 12, AS.
Yeah. You will still start AS Bio, or you already did?
Already started.
Okay, very good. So how many lessons did you cover?
We covered 1.5 chapter from Unit 1, and 2 chapters from Unit 2.
Okay, that's very good. So you already did a few lessons. Can you see my screen?
Yeah.
Okay, now very good. What's your real problem with AS?
I think answering the exam questions, like I understand the concepts, but in answering, like I lose a lot of marks, like not proper phrasing, maybe.
Not proper phrasing. Do you understand that you will sit for 2 papers?
Yeah.
We're talking here about Oxford, yes?
Yeah.
Yeah, you will be sitting for 2 papers, and these 2 papers are relatively straightforward. Both of them are short essay questions, and you don't have a practical paper like other examination boards. So that's a big advantage in Oxford. There's no practical paper. There's paper 1 and paper 2, a paper for Unit 1 and paper for Unit 2. You know that?
Yes.
Okay, very good. Now I have 4 participants here. We can talk, we'll just introduce the course, and we can start one of the easy lessons together for today. And then we will see how things will go. For the Oxford, you have 2 units. I'm sure you know this by now. You have Unit 1 and Unit 2. Both units are, the exam is 1 hour and 30 minutes. And the marks are 50% distributed over Unit 1 and Unit 2. And both units are 11 chapters. If you finish the first, like you started biological molecules?
Yes.
Okay, good. So
yes.
You're still here in biological molecules. You still did not move on to cells and cell structure.
I did 1 lesson in cells and cell structure.
Very good. Now, as you can see, there are 11 topics in Unit 1 and 11 topics in Unit 2. Okay. And the Unit 1, of course, is a little bit easier than Unit 2. You have here biological molecules. Here you talk about carbs, proteins, and lipids and their structures, and ATP and DNA, etc., etc. Unit 2 or Lesson 2 is very easy. It's divided into cells and microscopy. And you have enzymes, movement in and out of cells, diffusion, osmosis, active transport, and the structure of the phospholipid bilayer in cell membranes. You have the gas exchange system, and you have DNA and genes and inheritance in general. I don't think you started protein synthesis. It's too much into the syllabus. And DNA replication, meiosis, and you take mitosis also in cells and cell structure. So you will study both types of nuclear division, mitosis and meiosis, and classification, the last 2 lessons in Unit 1. And Unit 1, you have essay exam, no multiple choice, no practical paper in Oxford. Unit 2, you have here diseases and immunity. There's a nice unit over immunity. Digestive system, HIV is taking a big toll in the syllabus, and the circulatory system and the heart diseases and plants. And you have here mitosis again, mutation and cancer. So these are the lessons that you'll cover in Unit 1 and Unit 2 only for AS A level. You have Unit 3 and 4. Okay, so this is just a quick introduction over the units. Now, Miriam, you told me that you have a problem with answering the questions because the syllabus itself is not very hard. AS Oxford is relatively straightforward. You have a problem, you told me, with the exams and answering questions. Well, the exams for Oxford, let me open a sample of the classified. For example, this would be your classified. This is for the lesson cells and cell structure. Your classified will start from 217, okay? and it will end at the last exam here I have is January 2022. So the classified are grouped according to your lessons. There are not so many exams available for Oxford, especially the new syllabus. So I have the recent exams from 2017 to 2022, the past 5 years. And from those, I created classified from May 2017 until January 2022. Now, you will have the classified, the questions grouped according to topic, and you will have the answers. The answers will look exactly like this. This is the same classified for the lesson cell structure. You will have the question and the exam. Clearly, May 2017, the question, and the answer will be within the body of the question. So you will have the questions separately, and you will have the questions and the answers in another document. Do you understand how it will look like? Anyone has any questions? Hello? Can you all hear me?
Yeah, there's no questions.
Okay, so about the course, I introduced it quickly again for those who came late. You have 2 papers, paper 1 and paper 2 over unit 1 and unit 2. You have only 2 units in AS Bio. You don't have a practical exam. Both papers are short essay questions. They're clear, straightforward questions. You don't have a multiple choice paper. You don't have a practical paper like in other examination boards. These are the topics in unit 1. We will not go over them now. These are the topics in unit 1, 11 topics. And these are the topics in unit 2. Okay, so there are in total 22 topics. They're very easy and straightforward, maybe to me. The questions, because most of you have problems with answering exams, there are recent exams that I can get my hands on from 2017 until 2022, and you will receive questions per topic called classified. And I gave you a sample here. They look exactly like that. You will have clearly the title or the year from which this question came from. Like, for example, January 2022, this is over the lesson cell and cell structure. And in another document altogether, you will have the answer to the question. The answer will be within the body of the question. That question we were looking at. Here is the question, January 2022. If you compare, here you have the question empty with no answer, and here you will have the answers within the body of the question. You're supposed to solve these classified questions after every lesson. We finish a lesson, and then we solve these questions together to practice how the questions will come over that particular lesson. Okay? In my opinion, the easiest lesson is the cell structure. So we can start with that and understand how things will go. During our sessions, we will be using PowerPoints. And then when you are studying, you would use notes. These notes, they come from several different sources. They can come from internet, so they can come from a book, whatever. I group them together and I make notes. Notes over that part can look like this. I can show you a sample of the notes. For example, the notes can look like that. This is also a sample of the notes for the cell structure. You will have here in the header where you are at the moment, like this is section 2, lesson 1 and 2, cell structure and microscopy, and the body of the notes will look exactly like that. Okay? So for the lesson cell structure and microscopy, I'm showing you a sample of how things will look like. These are the notes. This is the classified for that lesson, the empty version. And this is the classified with the answers within the body of the question. And every other lesson will look the same. Clear? Anyone has any questions? Hello? Anyone wants to ask?
No.
Okay, we can start because I see that all of you already started the syllabus in school, so I can test your knowledge. We'll start with the easiest lesson, which is, in my opinion, cell structure and microscopy. And I know that you will already have lots of information over the cells and the organelles and the different organelles. So I can ask you, for example, let's talk about mitochondria. Who can discuss and tell me what is the internal structure of mitochondria and which type of microscope can we use to view it? Hello?
Can you use an electron microscope?
Okay, very good. Why would you use an electron microscope, not a light microscope?
Because an electron microscope has a higher resolution and a higher magnification.
Very good. Now, what is resolution?
Resolution is the minimum distance required to identify two objects as separate.
Very good. It is the ability to see two different objects as two, or the distance between two different objects. Okay, you said that an electron microscope has a higher resolution. And what is the resolution of the electron microscope? And what does the resolution of a microscope depend on anyway? So what's the let's take it step by step. The resolution of a microscope, whether an optical or an electron microscope, depends on what? What am I doing? Okay, very good. It depends on the wavelength of the waves being used in that microscope. If we're using an electron microscope, definitely we will use electron beams. Now, the wavelength of the electron beams, is it of a short wavelength or a long wavelength? High frequency or low frequency? Talk to me about the wavelength of the electron beams, or the electron beams in the electron microscope.
The electron beams have shorter wavelengths, so higher frequency.
Very good. Now, excellent. Someone said here that the electron microscope has high resolution. This is very good. You need to understand the term. If we are looking at electron beams and light waves, or electron waves and light waves, light beams, whatever. Okay. The electron beams, they look like that. And the light waves, they have a longer wavelength, okay, and less frequency. And the resolution is half the wavelength. What is the wavelength of an electron beam, and what is the resolution of an electron microscope? This is the wavelength of an electron beam, which is obviously shorter than that of a light wave, and it is of a higher frequency. If the resolution is half the wavelength, what is the wavelength of the electron beam, and what is the resolution of an electron microscope? Who knows the numbers? Okay, in case you don't know the numbers, I'll tell you. The wavelength of an electron beam is 1 nanometer, and the resolution of an electron microscope is half that, which is 0.5 nanometers. Now, why should you know the numbers? In case they ask you, or they tell you that two objects are, let's say, 5 nanometers apart, can you see them as two distinct objects or as one distinct object? What do you think? If you're using an electron microscope, can you see them as two or one object? Who will tell me? The resolution is 0.5 nanometers for an electron microscope. So if the two objects are more than 0.5 nanometers, you can see them as two. Yes? Are you with me?
Yes.
Okay, very good. So that's why you should know the numbers, and you should know every word you're using. When you told me that the electron microscope is of high resolution, you need to know exactly what does resolution mean and where it came from. The light microscope, or the optical microscope, is of low magnification and low resolution. The resolution of the light microscope is half its wavelength, and the wavelength of the shortest light wave is 400 nanometers. So the resolution is half of that. What will be the resolution of an optical microscope or a light microscope?
200 nanometers.
Excellent. 200 nanometers. So when they give you the size of organelles or the distance between organelles and they give you numbers, you will know which microscope you would use. You would use a light microscope or an electron microscope, depending on the size or the distance. Okay, maybe we dived into lots of detail. Let's go to the mitochondria that we were talking about, and your friend said that we will view it using an electron microscope. Very good. By the way, you can see a mitochondria with a high power light microscope, but if you want to see the internal structures, of course, you will use an electron microscope. Now, can you discuss the structure of a mitochondria? I'm going to remove the screen here, and someone will help me draw. I have a plain screen here, and I want to draw a mitochondria, and I want to label it. Let me start asking you questions. Will I draw a single layer or a double layer? Who will tell me? Is this a single layered organelle or a double layered organelle?
Double layered.
Excellent. So I will draw one outer layer and talk to me about the inner layer of the mitochondria. How will I draw it? Will it be a smooth layer or it will be folded? Who remembers? I'm not explaining here. I'm testing your knowledge. Okay, so in case you forgot, the mitochondria, the inner layer will be what? What am I drawing here? Is
Folded.
it folded? It's folded, yes. What are these foldings called? And what is their function? Who knows?
They're called cristae.
Yes, well done. They're called crista or cristae. And what is the function? What's the function of mitochondria in general?
Large surface area for aerobic respiration.
Excellent. Excellent. They increase surface area. Any foldings, if you remember the foldings also in the intestine, the microvilli, increases the surface area to increase the rate of the reaction. And what is the reaction that takes place in the mitochondria? You said it. I think your name is Maryam. It's aerobic respiration. And you have to say aerobic because there's another form of respiration. What is it? Who knows? There are two types of respiration.
Anaerobic respiration.
Excellent. Well done. Perfect. Which takes place where? In the mitochondria or in the cytoplasm of the cell?
Anaerobic takes place in cytoplasm.
Yes, well done. So let's go back to the mitochondria. When you come to study any organelle, the best way to study an organelle is to take a plain piece of paper and draw it. When you draw it, you will remember the organelle and you will label its structures and you will remember the function. So we drew an outer smooth layer, an inner layer that is called the crista. We remembered that its function is to increase the surface area for aerobic respiration. Who will remind me what is the inner matrix? Oh, I just gave the answer. Who will remind me what's the inner ground substance here called? I said it while I was speaking. Who noticed?
Matrix.
Yes, it's called the matrix. And it's easy to remember because which letter does the mitochondria start with?
M.
M, and matrix also starts with an M because we usually get confused between the matrix and the stroma found where? Who knows? In the
chloroplasts.
Well done. So it's usually a point of confusion. The stroma in the chloroplast and the matrix in the mitochondria. How will we remember them? In the mitochondria, it starts with an M and the matrix starts with an M. So this will help us remember that the matrix is in the mitochondria. Okay, what will I draw inside the matrix? There are some structures that I can add to the mitochondria.
Ribosomes.
Well done. Perfect. Ribosomes. Okay. Now these ribosomes are of which size?
Small.
Well done. You know exactly the size? 70 Vedbergs. Ribosomes, when we study them, we will know that they are of two sizes, the 70 Vedbergs and the 80 Vedbergs. The Vedberg is the rate or the speed at which it settles at the bottom of a test tube in a centrifuge. Don't mind if you don't understand that, I'll mention it later. But the ribosomes are the small-sized ribosomes. Excellent. What's the function of the ribosome?
They're the site of protein synthesis.
Perfect. Now, what else will I draw in the matrix of the mitochondria? I will draw something else. What's that? circular loop of what? The
DNA.
Excellent. Well done. So mitochondria has its own DNA. It's called mitochondrial DNA. So I want to remember, or I want to study the mitochondria. The best thing I will do is that I will draw. I will draw. Always draw in biology. Biology loves drawing. I will draw. Help me quickly. An outer smooth layer. And then what am I drawing here?
Foldings.
Yes, called what?
Christy.
Well done. What are these?
Ribosomes.
Quickly. Yes, what is that? The circular loop of what?
DNA.
Mitochondrial? Yes, well done. Mitochondrial DNA. Now, the significance of the ribosomes and the mitochondrial DNA is that the mitochondria can replicate on its own, regardless of the replication of the cell. And sometimes this is very important because some cells are more active than others and they need more energy, they need more ATP, especially muscle cells. So the cell itself doesn't need to divide. The mitochondria can replicate on its own because they have their own ribosomes and they have their own mitochondrial DNA. So they can replicate if the cell needs more energy. And the ground substance here is called the matrix. Very good. So we're done with the organelle mitochondria just because we drew it and we finished it. Now let's revise another one, which is close. This is the mitochondria here. The chloroplast. Again, I'm going to remove the picture and you will help me draw it. I will use green color. Why will I use green color? Wake up with me. This is which organelle I'm talking about here?
The chloroplast.
Okay. What is the function of the chloroplast?
It's where photosynthesis happens.
Very good. It traps sunlight, photosynthesis. Excellent. It is a single membrane organelle or a double membrane organelle. So how many membranes will I draw here? 1 or 2?
2.
2. Yes, it's a double membrane, but the inner membrane is not folded like the mitochondria. It is nice and smooth, like I'm drawing here. Okay, so it's a double membrane organelle. Now, what are these? You told me that you finished this lesson, so you should know. What are these?
These are thylakoids.
Excellent. They're membrane-bound sacs piled on top of each other. Every sac is called the thylakoid. Okay, it's called the thylakoid. Can you say thylakoid? They're easy, but the names need to be remembered. And there is another longer membrane-bound sac that joins the two structures here. Who remembers its name?
Lamella.
Very good, lamella. I only hear one person speaking. Maybe the rest did not take this lesson. It's okay. I'm not explaining. I'm just testing your knowledge. Put this in mind. So the chloroplast, number 1, it is a double membrane organelle. There is the first membrane here and the second membrane here. And we are looking at the inside of the chloroplast. It is made of membrane-bound sacs known as thylakoids that are piled on top of each other. Every group of thylakoids are called what? Who knows? Grana. Okay. So the whole pile, in case you're confused, is called the grana. And that pile is made up of thylakoids. And the grana are connected together by longer thylakoids known as lamella. Okay, a lot of names here. So again, let me draw another structure here. So I have membrane-bound sacs piled on top of each other. I'm doing it slowly so that you can understand it. Every one of them is called what? I want someone else to speak. Let me see how many participants. Okay. I don't have their names. I think Maryam was the one speaking. Malak. I have a Malak here. Hello, Malak?
Miss, his name is Malik.
Apologies. Malik. Hello, Malik. Okay, Maria.
Yes, miss.
Okay, Maria, can you tell me one of those membranes bound sacs, what is their name?
Thylakoid.
Thylakoid. And a group of thylakoids piled on top of each other, what is their name?
Grana.
Grana. Very good. And the thylakoids are connected together by a longer thylakoid called what?
Lamella.
Lamella. Very good. Excellent. Now, what else will I draw inside the chloroplast? What are these? Malik. What do you think they are? They are very similar to those that I drew in the mitochondria, and they produce proteins. Then what are they? Malik doesn't want to speak. Who will help him? Excuse me? Ribosomes. Okay, so these are ribosomes. Very good. Where's my picture? It's gone now. Okay, now this is the ribosome. Of course, my picture was much nicer. This is what I was trying to draw. If you notice, these are the two membranes. This is the first membrane and the second membrane because it's a double membrane organelle. Where's my pen? My pen is right there. So this is one membrane and another membrane. And these are the grana that we were talking about. They're made up of thylakoids. And when the grana are connected together, they're connected by lamella. Okay. And these are the smaller ribosomes. Now, which size are those ribosomes? The smaller or the larger ribosomes? Who knows? Hello?
Larger?
No. We will talk about the ribosomes in detail. These are the 70 bedbirds, or the smaller ribosomes. The larger ribosomes, my dear, are only found in the cytoplasm of the cell or on the surface of the rough endoplasmic reticulum. This is where they're only found. The rest, the remaining ribosomes that we will discuss in the mitochondria, in the chloroplast, in the prokaryotes, they're all the smaller size. They're all the 70 bedbirds. Okay. Now, this is where I stopped. I was going to draw here a circular loop of genetic material, and that also is DNA. So if we group together the organelles that contain DNA, it will include the mitochondria and the chloroplast. Again, why does the chloroplast have its own ribosome and DNA? For the same reason the mitochondria has its own ribosome and DNA. Why would a chloroplast has its own DNA and its own ribosome? Who will tell me? Girls. Hello?
Miss, the ribosomes is for the protein synthesis, but I'm not sure about the DNA.
Okay. Didn't we say that the mitochondria, they have their own ribosomes and their own DNA for a particular reason? What was that reason? If cells need more energy, does the whole cell divide or the cell produces more mitochondria?
More mitochondria?
Exactly. And this is exactly why the chloroplast has its own ribosomes and its own DNA, because the chloroplast can divide and make more chloroplasts independent of the cell division, regardless of the cell, whether it is dividing or not. If the cell wants more chloroplast because it wants to do more photosynthesis, the chloroplast can divide on its own because it has its own machines. It has its own ribosome and its own DNA. Okay? Is this clear?
Miss, so does the DNA control the activity?
Control the activity of the chloroplast. Yes.
Okay, thank you.
Okay, very good. Smart question. So again, quickly drawing and comparing the ribosome and the chloroplast. We usually compare them because they are similar. We have the mitochondria. I will draw it and you will help me label. Okay, so I will compare between, where's my screen here? I will compare between the mitochondria and the chloroplast. Okay, now if I draw inner foldings, this will be the mitochondria or the chloroplast?
Mitochondria.
Excellent. And these foldings, what are they called? Quickly help me label. Crista. Okay. Now, the inner matrix in a mitochondria, what is it called? I just gave you the answer.
The matrix.
Ground substance, yeah, it's called
Matrix.
Okay, excellent. What are these?
Ribosomes.
Very good. These are the 70 Vedberg's ribosomes, the smaller size, and this is the mitochondrial DNA. So we're done. Okay, now we'll compare that. I will draw another drawing. If you were with me, like if we were in a class, we would have drawn this together. This is a chloroplast. Can anyone draw? Who wants to draw? Can you draw on the screen with your mouse? Let me see.
Miss, I can draw. I'm using my phone and it's like hard to draw.
It's hard to draw, yeah. Okay, then help me draw. Okay. It's gone. Now I was drawing the chloroplast. Help me label. This is my outer membrane, and this is my inner membrane of a chloroplast, which is nice and smooth. Now, what are these? Help me label.
Thylakoid.
Very good. And a whole bunch of thylakoids piled on top of each other. What are they called?
Grana.
Excellent. Okay. Now, when they are connected together, these grana are connected by what?
Lamella.
Perfect. Lamella. Excellent. Now, these are what?
Ribosomes.
Well done. This is what?
Circular DNA.
It's circular DNA.
Very good. And the ground substance inside the mitochondria, it's not called the matrix. What is it?
Stroma.
Excellent. Well done. For your information, you will not take this in AS biology, but maybe in A level. The light-dependent reactions, they take place in the grana and the thylakoids because photosynthesis is divided into the light-dependent and the light-independent, in case you're asked. The light-dependent takes place in the thylakoids and the light-independent takes place in the stroma. Okay, so we're done with the mitochondria and the chloroplast. And this is a nice picture here showing you the chloroplast. You have the stroma here. Let me change color so you can see. And the thylakoids and the space between the two membranes, because this is a double membrane, the outer membrane and the inner membrane and the gram. Any questions over these two organelles?
No questions.
Clear? Okay. For your information, the chloroplast is larger than the mitochondria. It's larger. Hey, don't forget this. Okay, now let's discuss the ribosomes because you asked me about it. Now the ribosomes, again, I'll get a plain picture. What is the function? What's the function of the ribosome?
Protein synthesis.
Protein synthesis. If you notice how I drew a ribosome, it looks like a hat. Okay, why is that? Because the ribosome is made up of two subunits. A larger subunit, or a larger part, if you want to simplify matters, a large subunit and a smaller subunit, small subunit. Now the ribosome, you have to know some information about the ribosome. Where is it produced inside the cell? Do you know that? It's produced in the nucleolus. I'm sure you all know what is the nucleus, yes?
Yes.
Very good. Inside the nucleus, there is another body, another circular body known as the nucleolus. The ribosomes are produced inside the nucleolus. So this is where it is produced. This is the location of production. So ribosomes, number one, they are produced in the nucleolus. Two, their function. Of course, you know that they produce what? Proteins. Now, the structure of the ribosome. The ribosome is made up of two parts, a large subunit and a small subunit. And a very important piece of information about ribosomes, that they are non-membrane bound organelles. In other words, they are not covered by a membrane. They are not covered by a membrane. And that's very easy to remember because this is the only organelle that is not covered by a membrane. That is why it is the only organelle found in which type of cell? Prokaryote or eukaryote? Who knows? Do you know the difference between prokaryotic cells and eukaryotic cells?
Yes.
Very good. Prokaryotic cells, do they have a nucleus?
No.
Okay. Do they have membrane-bound organelles?
No, they don't.
Excellent. Then what is the only organelle that is found in the prokaryote? Look at the picture here. Ribosomes. Ribosomes, to cut a long story short and to prevent confusion, it's the only organelle that is found in prokaryotes because it's the only organelle that is not covered by a membrane. If you always read in biology books, they describe prokaryotes as cells or organisms that contain non-membrane-bound organelles. Did you read this before? While the eukaryotes, they contain membrane-bound organelles, organelles that are covered by a membrane. The only organelle that is not covered by the membrane is the what?
Ribosomes.
Is the ribosomes. Let's go and look at the structure of the ribosome. This is what I was trying to draw here. Okay. That ribosome is made up of two parts, as you can see, a large subunit and a small subunit. I always call it the hat. Fine. And it is not covered by a membrane, not surrounded by a membrane. It's the only organelle that is found in prokaryotes, and it's found also in eukaryotes, but it's the only organelle found in prokaryotes. Its function, it is the site of protein synthesis. And where is it produced? It is produced in the nucleolus. In case you didn't know, it's the body inside the nucleus. Okay? Ribosomes are of two sizes. There is a big size ribosome and a small size ribosome. Ribosomes, I think now you know where they're produced, their function, their structure. There are two sizes. You have the larger ribosomes and the smaller ribosomes. The larger ribosomes, where are they found? Who knows?
Eukaryotic cells.
Yes, you are correct. Where exactly in the eukaryotic cell? What am I drawing here?
Missed in the cytoplasm, maybe?
Yes, excellent. Ribosomes, they're either free, they're found in the cytoplasm, or they are what?
They're in the rough endoplasmic reticulum.
Excellent. They are attached to the endoplasmic reticulum. Where is the picture? This is the picture. This is the endoplasmic reticulum, and you have ribosomes here attached to the surface of the endoplasmic reticulum. And these ribosomes are of which size? These are the larger ribosomes. We call them the 80 Vetberg's ribosomes. In case you want to know what is a Vetberg, this is the unit. The unit at which how fast the ribosome or the organelle settles at the bottom of a test tube in a centrifuge. So going back to the ribosomes, we said they are produced in the nucleolus. They are not covered by a membrane. They're made up of two parts, the larger and the smaller subunit. And they are either of big size or smaller size. The big size ones are found either free in the cytoplasm or attached to the rough endoplasmic reticulum. Now the question is that where is the smaller ribosome found? Now we know the location of the larger ribosome. Where is the smaller ribosome found? I already mentioned two organelles which contains the smaller ribosomes.
The mitochondria and chloroplasts.
Excellent. Well done. And they're also found in prokaryotes. So let's group all this information together because we said lots of information. We're talking about ribosomes. Ribosomes. I'll ask you, you tell me, where are they produced? Produced where? Very good. Produced in the... in the nucleolus. Okay. Structure. Made up of what?
Two subunits, one large and one small.
Excellent. A large and a small subunit. Well done. And you can draw that. It looks like a hat to me. And larger and smaller subunit. Okay. And then we said function.
The site of protein synthesis.
Okay, the site of protein translation. If you know what translation is, you'll take it later. So it's the site of protein synthesis, and then location. This is the most important part. Now we said location? Yeah. Where is it found and what sizes? Who will tell me?
The larger one is present in the eukaryotic cells, in the cytoplasm, and the endoplasmic reticulum.
Perfect. Excellent. And the smaller sized, the 70 Vedbergs, where are they found?
Prokaryotics.
Excellent. And where else? They're also found in eukaryotic cells. Inside what?
The chloroplast and the mitochondria.
Well done. Amazing. Perfect. Good. So we're done with the third organelle, which is the ribosomes. Okay? Yeah?
I don't understand why there are two sizes.
You don't understand why there are two sizes because maybe the larger ribosome is too big to be inside the prokaryote. You have the bigger size and the smaller size, but they both have the same function. And the 80 Vedbergs will be too large to be inside the chloroplast or the mitochondria. Okay?
Okay, thank you. Yeah.
Welcome. Now let's talk about the organelle that is very close to the ribosome, which is the rough endoplasmic reticulum and the endoplasmic reticulum in general. Okay? Mind me, I like to draw, so I'm going to draw my cell and my, what's that?
The nucleus.
Yes. Okay. And what am I drawing here?
The endoplasmic reticulum.
Excellent. The endoplasmic reticulum. Okay. Now help me describe it. I was doing this. Can you describe the structure that I just drew here? If you're asked an essay question, describe the endoplasmic reticulum. How will you describe it?
It's folded.
Okay. What else?
They're flat and tubular.
Okay, they're flat. I have a question for you. Did I draw these foldings separately or they were connected?
Connected.
Connected. And this is the key, how we describe the endoplasmic reticulum. Because there's another organelle, and I'm sure you know about it, that is very similar to the endoplasmic reticulum, but it is made up of sacs that are not connected. Do you know which organelle I'm talking about?
Golgi apparatus?
Yes, the Golgi apparatus or the Golgi body. Very good. So let's focus on the endoplasmic reticulum and describing it. The endoplasmic reticulum is described in biology always using that word, interconnected. Interconnected membrane bound sacs piled on top of each other. Because when we describe the Golgi apparatus or the Golgi body, we will use the same description, but we will not use the word interconnected. Can you see the difference? The difference is in one word. In the endoplasmic reticulum, we will describe it saying they are made up of interconnected. Interconnected means they're joint. It's one big organelle. Interconnected membrane-bound sacs piled on top of each other. And these sacs, they have a name. What is the name of those sacs? Who knows? Cisterna. Can you say it?
Cisterna.
Cisterna. And this is usually a point of confusion between the crista and the matrix. The matrix has inner foldings called crista, But the endoplasmic reticulum and the Golgi body, they have membrane-bound sacs piled on top of each other called cisterna. What's the difference between the endoplasmic reticulum and the Golgi body? Is that in the endoplasmic reticulum, these membrane-bound sacs are interconnected, as you can see, they're one big organelle, but in the Golgi body, they're not interconnected. When you come to describe it in the Golgi body, you will mention or describe it that way, that it is made up of membrane-bound sacs that are not interconnected. These membrane-bound sacs are also called cisterna. So they're very similar. There is one small difference. Is that clear?
Yes.
Good. The endoplasmic reticulum is also divided into two types. What and what?
Rough and smooth.
Excellent. Well done. What's the difference between the rough and the smooth?
The rough
has
ribosomes on the surface.
Yes, excellent. Which size, which type of ribosomes? The small or the big one? The 80 or the 70 vertebrates?
The 80.
80.
Excellent. Perfect. And what's the difference between the rough and the smooth?
The rough has ribosomes on the surface, and the smooth doesn't have ribosomes on the surface.
Okay, talk to me about the smooth endoplasmic reticulum. What's its function?
It synthesizes, stores, and transports carbohydrates and lipids.
Okay, lipids, to be specific. Good. So the rough produces proteins and the smooth produces lipids. Okay, that's very good. We didn't, of course, finish the lesson. We're just asking some questions. I just want to look at the questions together. We can answer something related to what we took today. Now, these are the questions. I'll remove the mark scheme. Okay. This is a question from a 218 exam. Figure 1 is a drawing of a transmission electron micrograph of an E. coli bacterium. Now let's stop here. What is a transmission electron micrograph? Tell me. I forgot. If you took that lesson. Hello?
Yes, I didn't learn.
You didn't take it? Okay. Now for your information, the electron micrograph is of two types. You have the transmission and you have the scanning. The transmission, from the name, you can see the inside of the organelles or the structures. Okay. Now, can you help me label A and B and C?
Capsule is A.
Very good. A is the outer coating capsule. What's B?
Cell wall.
Excellent. Cell wall. What is C?
Cell membrane.
Cell membrane. Can you tell me what the cell wall is made up of in a bacteria? Is it made up of cellulose like the cell wall in plants?
No.
Excellent. It's made up of what then? Peptidoglycan. Maybe you don't know it. Peptidoglycan. Glycan or murin. Good. So your questions, your exams are actually very, very easy compared to the other examination boards. Let's look at the mark scheme because I want to show you how things would look like. This question. Okay, so you'll find the answers here in the body of the question itself. You have the capsule, cell wall, cell membrane, or cell surface membrane. Perfect. Your questions are easy, but the problem with the Oxford examinations is that their grading is very strict. So their exams are easy, but the grading is very strict. You have to give them exactly what they want. If you don't give them exactly what they want, they will not give you the grade. In other examination boards, you can write things differently in your own words, but as long as they have the same meaning, you will take the grade. You understand? But in Oxford, the exam is easy, but the answers, they have to be very precise. So the solution to that problem is that you need to practice as much as you can and to solve past papers as much as you can and look at the mark scheme so you know exactly how to write the answer and give the examiners what they want. Do you understand the difference here? Okay, let me see. Let me solve another question. Okay, let's solve this question. This is a specimen 218 exam. This is a microscopic aquatic microorganism called amoeba, a single-celled eukaryotic organism. Scientists use a transmission electron microscope to study an amoeba. Figure 1 shows its structure. Name the organelle Y. This is so easy. What's organelle Y? I'm zooming in. So
apparatus.
Y. Yeah, the Golgi apparatus. What is Z?
Mitochondria.
Mitochondria.
Tell me, what is the function of Y? They didn't ask you, but I'm asking you. Golgi apparatus, what is its function?
Miss, I'm not really sure, but it modifies proteins and packages them into vesicles.
Very good. Very good. Okay. We didn't explain the lesson. I'm telling you, I was just testing your knowledge. We did a few explanations. So yes, very good. What you said is actually very correct, that the Golgi apparatus, it modifies and packages proteins and lipids made by the endoplasmic reticulum, and the Z is aerobic respiration. Now, these questions are so easy to me because I would never find a question in AS Bio asking you to name an organelle. That's a question in a pre-IG exam. So your exams are relatively easy, but I'm telling you, the grading is very strict. Okay? Name 2 other structures in the diagram which show that the amoeba is eukaryotic. What other structures that you see here that tells you that that amoeba is an eukaryotic organism? Again, we did not study the difference between eukaryotic and prokaryotic.
Nucleus.
Yes, perfect. Number 1, membrane-bound nucleus. What else?
Membrane-bound organelle like the mitochondria.
The mitochondria. Okay, very good. The mitochondria is a membrane-bound organelle, and the Golgi body is a membrane-bound organelle, and the nucleus is a membrane-bound organelle. Now, membrane-bound, the term membrane-bound means it's covered by a membrane. And some organelles are covered by a single membrane, and some are covered by a double membrane. Now, to prevent confusion, those organelles that are covered by a double membrane are the large organelles, which are what? The nuclear membrane. This one, the nuclear membrane is a double membrane. The plasma membrane itself, or the cell membrane itself, it's called plasma membrane or cell membrane, plus the 2 organelles that we discussed today, the mitochondria and the what?
The chloroplast.
These are the 4 organelles that are covered by a double membrane. Anything else is covered by a single membrane, and the ribosome is not even covered by a membrane. This is the only organelle that is not membrane bound, and the only organelle found in the prokaryotes. Okay, now these small details are important. The organelles that are covered by a double membrane is the nuclear membrane, plasma membrane, this one, and the membrane of the cell itself, plus mitochondria and the chloroplast, if the cell has chloroplast, if it's a plant cell. And the remaining organelles, the Golgi body, the endoplasmic reticulum, the vesicles, the lysosomes, all these are covered by a single membrane. And the ribosome is not even covered by a membrane. Is this part clear?
Yes.
Okay, good. Let's solve one last question. Okay, what is the function of organelle Z?
It releases energy.
Yes, it releases energy. Now, this is what I'm telling you about. Okay, good. Let's look at the mark scheme. I'll tell you why I want to look at the mark scheme. Where is that question? This is the question here. Look. You had to say in the answer the term aerobic respiration, or you will not get the grade. This is what I'm telling you about, that their grading is very strict. Maybe because you did not take it yet, even though what you said is correct. But you did say ATP production. Very good. So what is the function of organelle Z? Z is the mitochondria. The mitochondria is the site of aerobic respiration, because later on you will study that there are two types of respiration, aerobic and anaerobic. The aerobic respiration takes place inside the mitochondria and it produces ATP. So in the answer, you had to either say the term aerobic respiration or the term ATP. If any one of those was not in the answer, you will not get the grade. This is what I'm telling you, that the grading is strict, but the questions are easy. Okay? Reject anaerobic respiration. If you wrote anaerobic respiration, then it will be wrong. If you just write to produce energy, it will be wrong because it's not enough. It's not a clear answer. It will be ignored. You understand? So your answers need to be precise. But even though, it's still easy. Okay, clear, girls?
Yes, miss.
Yes.
Does anyone have any questions?
No.
Okay. We did ask this question before. What is resolution? The distance between two objects to be able to see them. Now, this is what I was telling you here. Explain why electron, this is the first thing we explained in this session, why electron microscopes have a higher resolution than optical microscopes. Look at the answer. Let's look at the answer together. Electron microscopes use a beam of electrons that have a shorter wavelength and a higher frequency. Didn't we say that? Remember when we drew the electron beam? Okay. This was the electron beam and this was the light waves. And it was very obvious that the electron beam, they had a shorter wavelength. And if you have a shorter wavelength, this is a little bit of physics here, you will have more frequency. And we even mentioned more detail. What is the distance between the, what's the wavelength here? The distance of the wavelength of an electron beam. How many nanometers?
One?
Yes, you're correct. 1 nanometer. And the resolution, which is half of that, will be
0.5.
0.5 nanometers. In case they give you a distance and they want you to know whether you will be able to see that distance with an electron microscope or not. What is the wavelength in a light beam?
400.
400. And half of that?
200.
200. So the resolution of a light microscope is 200 nanometers. Okay? So your syllabus is easy and your exams are easy, but nothing goes without hard work. You need to study a little bit and it's easy to score a very good grade in Oxford. You don't have multiple choice, which is always the reason behind why students lose marks, because the questions are very tricky and you don't have a practical paper. But you do have practical questions, but you don't have a practical paper. You don't go and do a practical in a lab, because this is what they do in other examination boards, like Cambridge, for example. Okay, do you have any questions?
No, miss, thank you.
You're welcome. Okay, have a good day. Bye.
You too, Miss. Bye.
You too, miss. Bye.
Bye-bye.